Okay, so let's first of all review the Maillard reaction, and then we talk about meat tenderization. So we talk about non-enzymatic browning. Now there is two. So we talk about one of that is Maillard reaction. And we said the Maillard reaction is based on the Hodge-Maillard schedule. So Dr. Hodge did a literature review and pointed out how to explain it. So we can do some of the review, which means reducing sugar. Let's say you have a D-glucose. And we'll be, first of all, working with amino acids. Okay, any amino acids, or we can say any of the nitrogen-containing compounds, then become an substitute glucosamine. And then the second step, do some of the arrangement. We call it Amaduri rearrangement. Rearrangement will become 1 amino 1 deoxyketosamine. And this is like, we say it's a fructose, D-fructose. And then, depends on what type of the situation, we are going to different category. If the pH at 4 to 7, which means acid environments, they're going through 1,2 enolization. And will generate products called HMF, hydromethylfurfural. If the pH is about 7, let's say, or more than 7, we're going through 1,3 enolization. This will generate reductase. If the pH is more than 7, however, at a very high temperature, it could directly generate fission products or other volatile compounds. And then, if the reductase working with an alpha amino acids, they could be directly going through a process called Strecker degradation and will generate ammonium, carbon dioxide, and also aldehyde. And then all these products working in the food system with water, with imine, with other compounds, going through polymerization will generate a products contribute to the color is called melanoidin. That's why basically you see the Maillard reaction, it is brownish color. So this is what we go over before the exam. And when we talk about reducing sugar, and we mentioned reducing sugar, including like glucose, fructose, maltose, lots of that. Why we say it's a reducing sugar? Because it could generate a free CHO, which means a free aldehyde group. And the connected with that, we call it a hemiacetal. Now we mentioned that there is a one major carbohydrates is not reducing sugar, and everybody knows it is sucrose. Sucrose is not reducing sugar because it has a sido, and also we call it head to head connection. It's not head to tail connection. So sucrose is not a reducing sugar. There is no free aldehyde generated. Although sucrose is used to manufacture the table sugar. And we said that if the sucrose needs to go through Maillard reaction, it has to break down, become glucose and fructose first. Then they can go through Maillard reaction. So this is something we talked already. And we also have a slides we will go over next week, Tuesday. We'll go over next week, Tuesday, and also talk about the exam one. Now, we stopped here. We said there is another non-enzymatic browning. It's called caramelization. So what this means, which means in a food products, the reducing sugar or reducing carbohydrates directly reacted in a heated environment without a nitrogen containing compounds. However, they can react with sulfite and or ammonia. So if you look at this definition, there is clearly have 4 different categories of caramelization. Category number 1, we saw class number 1, is plain caramelization, which means reducing sugar at heating environments without sulfite nor ammonia. So what it looks like? What is this product? This is a product everybody will order. Sometimes it's a Starbucks coffee. So caramel coffee looks like that. And what's the color? It's brownish color. So that's class 1. Then what is class 2 caramelization? So if you look at the definition, which is reducing sugar in the heating environments with sulfite only. These type of the products, pH around 4 to 5, more accuracy is 4.2 to 4.8, and will create slight reddish brownish color. And what are the example? It's beer or other alcoholic drinks. So that can be reacted with sulfite. Now let's go to class 3 products. That is reducing sugar with heat, then reacted with ammonia only. And then we'll generate normal reddish brownish color, and the pH slightly lower, pH around 3.2 to 4.5 or 4.0-ish. What is the product is that? Syrup and pudding. Now, obviously, the class 4, you already know. It's reducing sugar with heating reacted both sulfite and ammonia and generate, we call it a dark reddish brownish color. No specific pH for that, pH value for that. What's that? Is cola drink, Coca-Cola or Pepsi. So that's very interesting. We can introduce you. When you order a drink from Starbucks, you have a Coca-Cola, you have a syrup to make the breakfast like pancake, or you have ordered the French pudding. Those are the products which is going through the process is another non-enzymatic browning process because of caramelization. Okay, so that's where we end the talk about this Maillard reaction and non-enzymatic browning. And then today's material, and also a little bit on Monday, we will talk about another topic, which is lecture 7. We will talk about is meat tenderization. This is a huge topic, and there's lots of the information there related to meat science, chemistry, and also slightly about the biochemistry. So we want to go over these things one by one. First of all, in the United States, basically, traditionally, there are two types of the beef products, which is forage feed, or sometimes we call it grass feed beef. Now what it looks like, the cattle is forage feed, so the beef is, we say, the grass feed beef products. What it looks like, it's grassy, it's oily, it is fishy. And people do some of the testing, find that the major lipids is linolenic acid. And we will learn this. C18, 3 double bonds, unsaturated fatty acids. Then the second category is, we know it's a general, common raised beef products, which is a grain feed. That means it's exactly the normal beef fat. So what are the components there? Like stearic acid, palmitic acids, all of that, but more important, they have oleic acids. That is C18, 1 double bonds, and linoleic acid. That is C18 double bonds 2, 2, which is 2 double bonds. So those are the two categories. Now, obviously, in recent years, they come out with like a cell-mediated beef, or like a plant protein-based beef products. If you go to Starbucks, you will see you can order like an Impossible meat, all those type of things. That's a different category. It's a manufactured products because they think we don't have enough resource for the protein, so we have to manufacturing something in the lab that is related to the topic is food sustainability. So that's not we're not gonna talk about something here, okay? So just want to let you the traditional forage feed, the grain feed. The second thing we want to talk about is when we talk about the meat quality. Now we have a lot of something we can test, like color, pH, water activity, moisture, fatty acids, amino acids, protein, all these things. But when we talk about the meat quality, we are talking about the quality. So there is three things people care about. One of the three items we care about. Number one is juice, is flavor. We all care about is that flavor. What's it tasted like? Second is juiciness. Is juiciness, how juicy it is, especially after cooking. The last one is tenderness. Then the American National Cattlemen's Beef Association, we call it NCBA, the acronym. Very similar to our Poultry Science or Animal Science Association. They have an annual meeting too. They did a survey around 30 years ago, in 1996, to see the consumers in the United States, what they care about if you order beef products. Then they find that there is about 38% of the people prefer flavor. There is about 13% of people, or 12% of people care about juiciness. But more than 50% of the consumers really care about the tenderness of the beef. And they are willing to pay even like $2 extra for 1 pound if they'll get more tender beef. Now, how are we going to measure the tenderness of the beef? There are lots of the methods, but traditionally, we can have two. Number one is we are measuring shear force value. That is called SFVS value. This is using a texture analyzer. We have in the lab, I'll ask Ian to do a demonstration, the shear force value, which means how many force you need to use to cut it or slice it. Because more tough, it's more energy and the force needs to be used. The second thing is we could do sensory evaluation. Like we did a lot in the lab, in Dr. Matak's lab. Now the sensory evaluation, we also could do two different categories. One is like a trained panel, which means you have like 10 people. The 10 people has been trained, pre-trained. And then they do the sensory evaluation. Another one is a consumer style. That means you randomly pick people. You may have asked 100 students in the Davis College to do the sensory evaluation. But they all using the evaluation score, we call it a 9-0-point hedonic score system. Okay? So this is the overall, we talk about the tenderness. Now, before we go to talk about the tenderness of the beef, we need to go through some of the basic knowledge about the muscle structure and also the microstructure of the muscle. Dr. Kenny's lecture already showed you the slides, but we want to talk about this in a little bit detail. So here is the thing. Let's say I have just drawn a little bit here. Okay, we have a bone connected with, or what we said with nerve. Then we have a artery, vein, or blood vessel, all these things, and a tender sometimes. We have a tender. Okay, then we come out with a muscle. A muscle looks like this, which is actually a bundle. So I always say the muscle is like this. So we draw a couple of circles. We draw a couple of circles like that. Okay. This is we call it a muscle. The muscle have a multiple layer and packaged with the membrane system. Now the first one, we call it MP myosin. The first one, EPI. Myosin. That's a muscle, the whole thing. Okay, now we're going to take one of them out. We're going to take one of them out. This draw a little bit small, a little bit large. Okay, so this we need to draw a little bit large like that. This muscle, take it out. This is a muscle bundle. The muscle bundle also has a membrane system there. Then the membrane system called perimysin. So here we call perimysin. Now inside, you also see this muscle bundle has lots of stuff there. Okay? Then we took this circle out. This circle we go here. What we call it? That is we call muscle fiber. Okay, when you have a muscle fiber, then you have two different types of the muscle, which is white and red. Now, obviously, you know, fish, chicken have a more white muscle compared to beef and pork. And lamb, they have a beef, pork, lamb, they have a huge amount of the red muscle fiber. And then this is also packaged with a membrane that is called endomysin. Okay, then this muscle fiber, they also have multiple these layers. Okay, then they go out. We go out. And we can draw like this. Then what do we say? This name called sarcolemma. And sarcolemma had a very important components. Sarcolemma. Sarcolemma has a very important inner structure called sarcolemma reticulum, acronym SR. This is a place in storage calcium. This is a key component for muscle contraction and muscle relaxing. So we say SR. And then we also going to see there is some of the bundles here. Okay? Endomysin. We have epimysin, perimysin, endomysin. Then we going through these. We call it myofibril. And then they all come out another very thin stuff. We call it myofilament. And then the myofilament, we obviously know there's two kinds, which is myosin and actin. So this is a picture which is showing you the muscle is a bonding system with multiple layers. Now the scientist, the meat scientist back in 1930s, they using a very old microscope. They just cut the cross section of here. This is called a small section right here, okay? Sarcomere. Sarcomere, they think it is a single unit of a muscle structure. So we need to talk about what is sarcomere looks like. Therefore, we need to go through some of a little bit more detail. So the sarcomere, what it looks like. We're going to draw a couple of lines first. One line is here. One line is here. There is another line which is right in the middle. Okay. First of all, in the middle, we call this is A band. And outside, we call it is I band. This is I band. This is A band. Here is I band. Okay, what does I means? This I is isotrophic. So what this means? Refractive or refraction of light. And then what is A band? That is called anisotrophic. So not a reflection of the light. You can understand about that. Now the center of this line is we call M line. And the side of this line, we call it Z line. Then, if we talk in more detail, the A band have couple of zones. Right in the middle, this here, we call it H zone. And some of the textbook, if they go much more detail, they will say during the H zone, right in the middle here, there's a small section they call pseudo H zone. Okay? Now, what is so-called sarcomere? Sarcomere is the distance between Z line, the two Z lines. This is we call sarcomere. It's a single unit of a muscle structure. Okay, then we have these, then what else we have? We haven't talked about the myosin and the actin. What are they looks like? So what are the myosin actin looks like? So we're going to draw. It's very interesting, we can draw it. Okay, so we have a couple of things like this. Relatively thick. I'm not going to draw everything. Okay, relatively thick right here. So we have relatively thick things right here. And we're also going to draw something, a couple of things right like here. Okay. What are these? You draw like that. It's draw a little bit thick. So what do we call? That's thick filament or myofilament. How thick it is? It's around 14 to 16 nanometer length, nanometer radius. And it is about 1.5 micrometer length. And then on the top of here, you will see lots of things like that. It's like a sprouts. Lots of the stuff like this. Attached to the thick filaments. So we can draw like that. Draw a couple just. So what are they? So let's draw first, then we talk about it. All these like a sprouts. This is called mousing. So on the top here we can goes is mousing. Okay, now we're going to talk about mousing. What is mousing? Mousing is structure protein. So we haven't go through this yet. So muscle protein, there is two parts. Structure protein and water soluble protein. Now obviously structure protein cannot dissolve into water, otherwise we get a problem. So structure protein around 70%, water soluble protein is about 30%. And structure protein, there's basically four types. One is myosin, is 38%. Then another one is actin, is about 13%. Then we have another one called tryptomyosin. That is about 7%. The last one is we call it stroma. It's about 6% only. Okay, so myosin, around 38% is the muscle structure protein. The molecular weight is very high. It's 500,000 molecular weight. We call it delta. And around 60 to 70%, the second structure is alpha helix. So it looks like this. It's alpha helix. The major components of this protein, the amino acids, is aspartic acid and glutamic acids. So what are they? Aspartic acids. So let's just do it. COOH, we go CH2, we go this, we go NH2. That's aspartic acids. What's glutamic acids? NH2, COOH, H, we go CH2, CH2, this guy here, NH2 right here. Okay, glutamic acids and aspartic acids. Now, very importantly for this mouse, we need to know an important thing for mouthing. Mouthing have an important enzyme, which is ADP. Can be using a phosphate become ATP. That is, we call myosin ATPase. This is important, can contribute for ATP, because ATP is important for generate, needed for muscle relaxing and muscle contraction. You can go the other way, okay? We also want to mention this myosin ATPase is rely on a subunit of a myosin. We call it a married myosin. The married myosin, the molecular weight is about 220,000 delta. So it's huge. Okay, that's myosin. Okay, what are we missing here? I'd like to draw the picture. So what are we missing here? We have other ingredients is important, is something like Using a different color. We have a red color here. Okay, we have one. What else we have? Something like this existing. So one is goes there, another one is goes here. Then one is goes here. There's one goes here. And they also can be connected. Okay. What else is this red one here? So we do connection. We do connection. What is this guy? Acting. Okay, so we're going to talk about acting. What is acting? I'm going to draw on the top. Actin, major components of thin filaments. So that's thin filaments. How thin it is? Thin filament, around half the radius of the thick filament. So 6 to 8 nanometer radius, and around 1.0 micrometer length. And the actin, we already talked about, it's around 13% of a structured protein. The major, there is two different types of the actin, which is G actin or F actin. That indicates globular and fibrous actin. And the molecular weight is relatively small. The molecular weight somehow is between, we say like 18,000-ish, not very high. It's only a small piece section of those myosin, the molecular weight. So we know the ratio of actin myosin is basically 3 to 1. And what is amino acids inside of actin is proline. So we go back to see what is this guy. We need to draw the whole thing. CHCOOH. You have a CH2, you have a circle CH2, you have a CH2, you go here. Okay? So that's actin. Okay. Not only that, you don't forget about surrounding this xenon, there are lots of other proteins surrounding this xenon. What are they? That's numerous. Could be called nebulin, desmin, and titin. There are a lot, and etc. Maybe like 8 to 10 kinds of the protein are surrounded there. Okay. This is important. The background information related to the muscle structure, related to meat tenderization. Okay, now, before we go talk about tenderization, there is something we want to emphasize here, yes. The muscle contraction and relaxing. This you learned in your animal science class a little bit. So we want to emphasize here because the knowledge is related to that. So first of all, muscle contraction. They needed to use ATP. So ATP will be hydrolyzed, become ADP, and phosphate. This needs an enzyme, which is myosin ATPase. Of course, they need calcium, they need magnesium, all those type of the chemicals. Muscle relaxing, go the other way. which means if muscle do relaxing, they have to create and generate ATP. This sometimes rely on creatine phosphates. Now we should know that in the normal life, the contraction and the relaxing, you do this, contraction and relaxing has to be reversible at every second. Otherwise, the animal is dead. So, which means the body should generate ATP, hydrolyze ATP naturally at every second. Now, there is a little bit of chemical between that. What the chemistry behind it is major thing is the calcium. So, what is muscle contraction will be happening? During the muscle contraction, they have a very important neurotransmitter is acetylcholine. CHLOO is acetylcholine. Okay, this acetylcholine is a neurotransmitter. They will give the brain a signal, okay? We have to do muscle contraction. Once this signal transferred there to the motor plate, let's say, what will happen? The calcium will release from SR, which is sarcolemma reticulum, and go to sarcolemma plasma. Okay, once calcium is released, what will happen? The calcium will connected with a chemical called troponin C. Once they connected with troponin C, they will be trigger the tropomyosin to change the configuration. Then the tropomyosin will present or presenting myosin connected with actin and forming actin myosin cross bridge. This is important, which is how they connected. Right here, how they connected. So if you want to draw a little bigger, that's how they connected. That's actin myosin cross bridge. This is the key thing here. Related to the future, we talk about tenderization. Okay, what is relaxing? Relaxing has another neurotransmitter, a chemical called chloride. The another chemical is called relaxing is chlorine esterase. It's an enzyme, esterase. Chlorine esterase will break acetylcholine. So what happened? Calcium back to sarcolemma reticulum. So what happened? We'll stop the actin myosin cross bridge because they basically stopped here. The tropomyosin no longer presenting actin to present to myosin, so there's no cross bridges generated. Now, by the way, we talk about in the class, in the microbiology class, acetylchlorine is a transmitter. Chlorine esterase is also a transmitter. there is a toxin could block it, which is bacteria, Clostridium botulism. Is that right? So, muscle contraction, Clostridium botulism. The botulism toxin going to block here. So what happened? You cannot do contraction. You can always keep it relaxing. What happened? You have a floppy baby. Remember I talk about the joke? I said never feed a 1-year-old young baby honey, because honey is dominant with Clostridium botulism. They will block acetylchloride, so you have a floppy baby, which means the muscle is always relaxing. And in other words, there's another bacteria, Clostridium tetani. The Clostridium tetani is blocks what? Chlorine esterase. So the muscle is cannot relaxing, it's always contraction. Then what happens? You have a locked jaw. So that's what we learned in your microbiology class. But here, we want to mention here, because the idea here we want to show you is this calcium is extremely important. Control the calcium amount and where they go is important. Another thing is this ATP. You always should have an ATP reversible, which generated and depleted, always should be keeping a balance. If one of the balance is not kept, then the animal will be die, and the muscle will not keep the normal status. So that's why I want to explain to you here, because calcium and ATP are the two things are important. Okay, now we get to the next point. We may not gonna finish today. We're gonna continue to talk. Okay, number 5, what we got. We need to know a basic chemistry behind a cattle carcass to be a meat. There could be a 1 month or a couple of lessons in principles meat science class, but we just want to be brief. So we had a carcass, a cattle carcasses. We do slaughter, which means we kill it. So what happened? We have a bleeding out. We're bleeding it out.